Goeienaand, bonsoir, dumelang, goedenavond, good evening, and a very warm welcome to you all to Professor Edkins' professorial inaugural address. I extend a very, very special welcome to the family and friends, as well as collaborators and colleagues of Professor Edkins, some of whom have joined this special university celebration via digital platforms. This evening, as is our tradition, we have the presentation of an inaugural lecture, which follows the university conferring the status of full professor on an academic. This time-honored tradition, with origins in the medieval university, serves as an introduction and an induction of a new professor, and as a platform to showcase their scholarly contributions in their discipline. It is also an occasion in which we as academic peers, colleagues, students, family, friends, and the public celebrate the intellectual and scholarly achievements and contributions of one of our own. Promotion or appointment to the rank of full professor marks a significant milestone in an academic's professional and intellectual career. On behalf of the Rhodes University community, I offer our heartfelt congratulations, you Professor Edkins, on your promotion to the rank of full professor and for reaching this pinnacle of your career in the academy. Honored guests, this evening I have a singular honor of presenting to you one of the newest additions to the illustrious community of scholars who hold the rank of full professor of Rhodes University, Adrienne Edkins. Born in Bulawayo in Zimbabwe, Adrienne completed her education at Arundel Senior School before reading for her BSc in biochemistry and microbiology at Rhodes University. She started her postgraduate studies at our university, completing her honors in biochemistry under the supervision of Professor Stephen Burton, and her master's MSc in biochemistry under the supervision of Professor Greg Bletch. In 2003, she was awarded a Byte International Fellowship to complete an MSc in forensic science at King's College in London, where she worked with Professor Virginia Murray at the Division of Chemical Hazards and Poisons of the UK Health Protection Agency. This led to a prestigious Wellcome Trust PhD fellowship at Glasgow University. During this time, she completed a Master's of Research degree in molecular basis of disease before joining the laboratory of Professor Bill Cushley for her PhD in cellular biochemistry. After graduating in 2008, she returned to Rhodes University to conduct postdoctoral research with Professor Greg Bletch. In 2009, Professor Bletch founded the Biomedical Biotechnology Research Unit, BioBrew, with initial funding from Rhodes University, leading to Professor Edkins' appointment as a contract lecturer in biochemistry. This opportunity marked the beginning of Professor Edkins' journey within the academic ranks, advancing from a lecturer in biochemistry in 2009 to a senior lecturer in 2013. In 2015, Professor Edkins was honored with the South African Research Chair, a SACHI Chair, from the Department of Science and Innovation and the National Research Foundation. This prestigious chair provides her with the opportunity to devote her efforts exclusively to research and postgraduate training. With this support, Professor Edkins has established a thriving research team and has devoted her focus on her scientific endeavors, leading to the successful acquisition of several distinguished research grants. Notably, she secured 2 significant equipment grants from the National Research Foundation. She also secured an esteemed Advanced Newton Fellowship from the Academy of Medical Sciences in the UK, and a prestigious Trekanto Open Lab Fellowship in collaboration with the pharmaceutical giant GlaxoSmithKline. Continuing to excel in her field, she further progressed to the rank of associate professor from 2016 to 2022, before attaining her current esteemed academic rank of full professor of Rhodes University. Throughout her career, Professor Edkins has earned numerous awards and achieved remarkable distinctions in her academic and research endeavors. In recognition of her outstanding contributions to her field of research, she received the DST South African Women in Science Award, SAWISA, in the category of Distinguished Young Scientist in the Natural and Engineering Sciences in 2018. Of course, she also won the Vice Chancellor's Distinguished Research Medal in 2015-2016, and the Rhodes University Merit Award in 2014 for her research and teaching excellence, which has been acknowledged broadly. She's an NRF-rated scientist, an elected fellow of the African Academy of Sciences, and a member of the Self-Stress Society International since 2020, and a member of Academy of Science of South Africa, this since 2017. Demonstrating her continued excellence, Professor Edkins recently achieved a remarkable recognition by being among the select few to receive one of 4 grants awarded in 2022 under the esteemed UK Research and Innovation, UCRI Medical Research Council African Research Leaders Scheme. Professor Edkins has maintained a consistent presence among the top 20 highly productive researchers at Rhodes University. Her extensive scholarly contributions include authoring more than 70 peer-reviewed journal articles, contributing to 9 book chapters, and co-editing 4 books. Additionally, she has been an active participant in numerous national and international conferences. Notably, Professor Edkins has also played a significant role in shaping the academic landscape by mentoring and guiding 30 PhD students and 35 MSc students. Professor Edkins has actively engaged with the community through a diverse range of activities, demonstrating her commitment to promoting science education and awareness. In 2023, she participated in the Academic Exposure Program at Rhodes University, conducting interactive biochemical experimental workshops for local school learners to showcase research in biochemical and molecular sciences. Over the years, she has served as a judge for the Rhodes University ESCOM Expo for Young Scientists Regional Final, assisted with science journalism for a health block and contributed to SciFest events by organizing interactive workshops and providing information on DNA and forensic science. Professor Edkins has been involved in various initiatives, including cancer awareness initiatives at Rhodes University and the Science for the 21st Century lecture series. She was a member of the organizing committee thereof. However, despite all her remarkable achievements in the academy, Professor Edkins maintains that her proudest moment was the birth of her daughter, Renee, in 2016. Now, colleagues and friends, it is my singular honor to invite Professor Edkins to address us on her research into understanding the molecular mechanisms that control protein folding and homeostasis in health and disease, and how this understanding can be applied to identify new approaches to treat diseases. Professor Edkins.
Thank you. Okay, I'll take this. Can you hear me through the mic? Thank you very much for that wonderful and comprehensive introduction. No pressure. So it's sorry, I should actually get the presentation going. You want to do that for me? Okay. Okay, so thank you very much to everybody who's made the effort to be here tonight. It's a great privilege for me to have you here and to be able to have a captive audience to speak on my favorite subject. I have designed my talk in sort of 3 sections. The first is basically a short reflection on my journey. Then I will talk a little bit about the area in which we work and why we feel it's important. And then I'll end off with a little bit of information on some specific work that we've done. In terms of the first section, obviously it's a very personal reflection, and coupled with this microphone is probably going to come across as a, version of an Oscar acceptance speech. So I really hope you will indulge me and allow me this opportunity, primarily because I want to use it to thank the people who have who've really helped me along the way. Because I'm sure everybody knows, no matter where you are in life, there's no way that anybody got there alone. And so it's a wonderful opportunity for me to be able to say thank you through this reflection. So tonight, and the promotion is doubly important to me because I'm a graduate of Rhodes University and because I believe strongly in Rhodes University and what Rhodes University stands for. So it really is a double privilege. I arrived here about in 1997, the week before O Week started. Now, those of you who live here permanently or even temporarily will know that that's the week when it's so quiet, there aren't even any tumbleweed in town. And I thought, what have I done? And of course, little did I know that this would then become my home. And in fact, that week of the year would become my favorite week. Needless to say, I went on to make very many special friends and people who are still part of my life. Obviously, I met Gary, my partner here, and also a great group of friends from our days in Athens. We recently met up, we're still in touch. But perhaps scientifically, the first influence on my life was Petra Gens, Dr. Petra Gens. She's now the deputy head for academics at DSG, so I should probably watch what I'm going to say. But I'd originally arrived to do a BSc IS And luckily sense prevailed and I moved over to biochemistry. I don't think I would have been great inflicting myself upon the IS department. And in second year, I met Petra. And Petra was this, and still is, amazing, phenomenal tour de force personality. And we hit it off straight away, not least of all because Petra was very protective of her lecture notes. And so if you would ask her to borrow them, she would automatically produce free printed copies. This was back in the day before Are You Connected? And so you can imagine that that was invaluable. Petra was also very cool because she had a Nokia 5110 phone. This is the variety that you still had to pull the aerial out when you answered it. And it gave us an excellent business-like appearance, even though we were only in second year. And I knew we were going to be friends for life when she gave me that phone when she upgraded. And it caused quite a stir that Christmas in Zimbabwe. So we would go on to be lab mates and work our way through undergrad and stay on for postgraduate degrees. And it's wonderful that she's still in Grahamstown and I can still call her amongst my special friends. And then I did some of my postgraduate training at Rhodes as well. So I worked with Prof. Steph Burton. And again, that was a really great introduction into biochemistry, particularly enzymology. And then did an MSc with Greg, who it's wonderful that he could join us here tonight. And after that, I decided that I wanted to go overseas, and I was fortunate enough to get funding from the Bike Trust to allow me to go to London and do forensic science. And there I worked with Virginia Murray, who's behind that picture. But anyway, I worked with her, and she was this formidable woman who was constantly worried about what was going to happen in London and if there was going to be these outbreaks and whatnot. And we worked on a very exciting project trying to figure out what you would do with contaminated people if there was ever an attack. And it was a very exciting time and whatnot. But I learned very quickly that it wasn't CSI and that I didn't have the stomach for it, particularly after the time of death practical in the hottest summer they'd had in 20 years in London. That really focused my attention. So from that point, I again was lucky to get funding from the Wellcome Trust, and I moved to Glasgow. This picture is probably symbolic of how I spent most of my time, with my head down, whether it was working or climbing a mountain. But it was, again, a really wonderful experience because we were 9 students from 8 different countries on a joint PhD program, and not a Scottish person amongst them. Not that that's a bad thing, but what it meant was that we all were invested in each other because we were all foreigners and we were all together in this environment. And again, I've stayed good friends with many of these people who now have their own laboratories. And I worked there with Bill Cushley, who's now the Assistant Vice Principal International at Glasgow University. And an interesting story. So, you know how we always think that things are better and cleverer somewhere else. So when I was in Scotland, my PhD, I participated in a program called Biotechnology Yes, or Young Entrepreneurs Scheme. And this was really like a business pitch. So you had to come up with a science or biotech-based project and you would pitch it. And we went with the project that I had done at honors level in Steph Burton's lab. And we made it through to the finals on this project. And at the finals, we met another group who had pitched an idea that had come out of John Duncan's lab. And so it just happened that in the whole of the UK, in that year, 2 of the final projects in this Biotechnology Yes project had their academic origins and their intellectual origins at Rhodes University. And that's something that we can really be proud of. And yes, we won the competition. So that was wonderful. So then after 4 bordering on 5 years of being cold and damp and not understanding what everyone was saying, I decided it was time to come back home, and I really wanted to use the skills that I gained overseas to come and build biochemistry here. And so I was very fortunate that I was able to get back in touch with Greg, and he had a postdoctoral position available at the time. And so I came back to Rhodes University to join as a postdoc in 2008 after I just finished my PhD. And at that time, Greg, on the basis of Greg's productivity, had negotiated an investment from the then Vice Chancellor, Dr. Salim Badat, to set up the Biomedical Biotechnology Research Unit. And that really was, part of that was money for the salary that allowed me to then essentially start my own laboratory, and join first on a temporary basis and then on a permanent basis, the department in which I'd done my undergraduate and postgraduate training. And so here I should really just say thank you to my colleagues, some of whom were teaching when I was a student and are now my colleagues. I don't know if they're vampires or something. I'm assuming some of them are going to retire one of these days. But they really have been amazing to me, and I haven't felt like I was a student returning to my old department. I felt like I was taken seriously and really engaged. And we're like a family, I think, warts and all, I think, in that sense. You know, it's not always plain sailing, but we motivate each other, and we work very effectively as a team. And then from there, I should obviously say I've been able to build from there a group of collaborators, including people in the department. So Garth and Brendan and Brett, who've been very kind to me and very supportive. David Kanye, who is now in pharmacy. And then collaborators outside of the department, so Clint Veal and Carmenita. And then my colleagues in Rubik, who again have supported me either through collaborations or just being there for mentorship. And then, as usual, the most important person on that slide is hidden by the thing. So that's Joanne Delamere. So Jo started out as a PhD student in the group, and I was fortunate enough to be part of her supervision team. And so she has been working with me for many, many years and has been a great support. And I've really enjoyed that from her. And obviously, I'm sure there are many other people who I've forgotten to point out. So please, forgive me if if there are any omissions, inadvertent omissions. It's not the case. And then obviously, I should just acknowledge the role of the leadership at Rhodes. The past leadership when I first started, Dr. Badat and Professor Rick Bernard, but also the current leadership for creating an environment where I feel that I've had opportunities that I would never have had elsewhere. And I really feel that Rhodes is an enabling environment. I think we can be that way because we have agility in terms of our size. But just because you can be that way doesn't mean you always are. And so I'm really grateful for that. And in particular, I'm very grateful to Peter to say thank you for supporting me, in particular around the Saatchi chair. I was very young and precocious, perhaps, when I went there and I asked to be considered for that, to be put forward for that position. And I recognized that had I been somewhere else, I may have not even had an opportunity to put in an application, never mind be successful. And so I really appreciate that positive attitude because I think it's made all the difference. And then, of course, last but not least on this slide, I have to really say a huge thank you to Greg. I tried to look for one of those pictures of him in his old anorak. I'm sure everyone who's in the department around my time will remember he got the anorak award from the undergrads about 3 times in a row. So I had to settle for a pink shirt. He's gone very corporate recently, so I've had to dredge out the old photos. But Greg has been such a consistent, stabilizing force and an amazing mentor to me. And I really, really cannot thank him enough for everything that he's done, because I think it's been a make or break relationship for me. And I really appreciate it. And again, testament to his commitment. I appreciate that you made the effort to be here on this evening, Greg. Thank you. And then, of course, just to say, you know, thanks very much, to acknowledge the role that funders have played. Obviously, the work that we do is experimental sciences, so it's really expensive. We need to have resources available that allow us to buy chemicals, etc. And so the ability to access funding has been really, really important. And again, this is where the Saatchi chair, which is a research-only position for those people who are not familiar with it, and is a 5-year funding scheme, renewable for up to 15 years. And that kind of continuity has really made a huge difference in terms of being able to then leverage that in terms of both local and international funding. And I'm really grateful to the funders who've supported our work. And then last but not least in my Oscar acceptance speech is to say a huge, huge thank you to the postgraduate researchers and postdoctoral researchers who have worked with me and have been part of the team. This really is what it's all about for me. This is where I derive the joy from my research, because it doesn't matter if you have a great result in science, if you have nobody to get excited with over it. And it's really been a privilege to work with some of the most clever, motivated, and supportive students that anybody could ask for. I hate it when they leave, so my favorite ones are always the ones that stay around for longer. And I always tell them that they're welcome to come back. And in fact, I really want them to do that. But I'm also obviously proud that so many of them have gone on to do amazing things in science themselves. They've gone on to start their own labs in academia, to be part of biotech. There's one that even became a comedian. And I'm not sure I had a role in that, but anyway. And so I say a big thank you. Without them, none of this would be possible. I really am able to stand up here today and receive this wonderful acknowledgement because of their hard work and contributions. And so I really, really say thank you very much. I appreciate everything. And lab opens 7:30 tomorrow. Joking. Okay, so now I'm going to change tack a little bit, and I'm going to talk a little bit about the broad area in which we do our research. Okay, so we are biochemists or cell biologists, and basically we work at the level of the cell. So the cell is what you would consider as the smallest functional unit of an organism. Okay, so you have about 30 billion of these in your body. All right, and we study the processes that support viability of those cells at a single cell level, which then we can apply to organism level. And essentially, the cell is controlled by DNA. So if we think about that as the recipe book, that's the recipe book to make your cell. It contains every recipe you might possibly need in your entire life. You then can go from DNA, you can make a copy of certain recipes. So let's say it's breakfast time, you feel like making a certain recipe, you can extract that in an RNA form, okay? And then you can use that RNA to make a protein. And the protein is basically equivalent to the meal that you would make from that recipe, okay? And essentially, most people tend to go on about DNA. So there's been a lot of interest in DNA sequencing, things like that. We know a lot about RNA recently because of the COVID vaccine, which was RNA-based. But I'm here tonight to really advocate for the protein. All right, I really believe that this is where it all happens. And this is really the extent or the focus of my group is that protein. So if you consider that a single cell, something like this, will have around 42 million different proteins, okay? And that's squashed into a space about 100 times smaller than a human hair. Okay, so you can imagine that this cell is absolutely jam-packed with proteins. Okay. And the importance of proteins can be demonstrated if we look, for example, at all of these examples of different types of cells in the body. Okay, so we have some skin cells, muscle cells, blood cells, nerve cells. All of those cells have the same DNA. Okay, if they come from the same person, they have exactly the same DNA. But what's different about them are the proteins. So proteins really control critical functions in those cells, and they're important for specializing cells for particular types of activities. And obviously here are some of the different roles that these proteins might play. If we think about protein, we probably think about what, a steak maybe, or an egg, okay? So these are obviously ideas of proteins that are in common knowledge, but actually proteins are far more beautiful, far more intricate, far more detailed if you look at them at their molecular level. So here's just an example of some particular types of proteins, just to show you. You can see the intricate shapes that they form. So this protein here is responsible for getting glucose into your cells. We'll all use that in about half an hour. This protein down here is called rubisco, okay? Arguably the single most important protein for life, because that molecule is responsible for fixing carbon fixing carbon dioxide in photosynthesis. And so it gives rise to all plants, and hence life can originate from there. We have obviously a garbage disposal, which is the proteasome. You can see again, massive complexes. And then I have to move this one now because we can see here's the dustbin of the cell. And then this tiny little thing over here is the cobra toxin. And so it's amazing to think that something like this can have dramatic effects on an organism, even though it is fairly simple if you compare it to something like that. This here is ricin, which is a well-known toxin as well, and hugely toxic at very low levels. So all of these proteins have this specific three-dimensional structure, but they are not produced like that. Even though all of these proteins are different, they look different, they specialize for specific functions, they all have a common problem. Okay. Oops, sorry, beg your pardon. Oh, I forgot about this one, sorry. Before I moved on to that, proteins can also have really neat functions. So have you ever wondered why a jellyfish will glow? That's because of a specific protein, very creatively named green fluorescent protein. That protein does what it says on the tin. But it's also an interesting protein. When I used to lecture undergrads, I used to tell the story of the Nobel Prize for 2008, where these three individuals won that Nobel Prize for discovering green fluorescent protein. But what's interesting was they didn't actually discover it. This guy, Douglas Prasher, discovered it. But he shared the gene that was used, that he made for that protein with these three, and then he promptly left science, and he's now drives a courtesy car for some company in America. These three people went on to do a substantial amount of work and to win the Nobel Prize. But it's okay because they invited him to the ceremony. That normally gets a huge reaction. People hate them after that. And what's also interesting is with the ingenuity of humans, we now have a whole bunch of varieties of this protein, where if you change very few residues or very few positions in that protein, you can end up with this range of fluorescent proteins of different colors. Okay, and they're called the M fruits. Okay, M plum, M grape, etc., etc. The hardest one to work with is M apple because you're never quite sure whether it's red or green. But all of these proteins have one thing in common. Okay, they have what is known as a folding problem. They have an origami problem. Because they're made in a form like this, they're synthesized or produced in the cell as chains of amino acids, like beads on a string, but they're not active in that form, okay? And so they have to fold into those intricate, beautiful three-dimensional structures in order to be active. Now, the information to get here is in that protein sequence, okay? So it's determined by the chemistry of those amino acids. But the process of getting here is far more difficult. And often it doesn't always happen well. So proteins, by their nature, by the fact that they have this transition between unfolded and folded, are dynamic molecules. And so they are susceptible to what we would collectively term as stress. So these are either changes in environment or they are mutations. Okay, so mutations would change this sequence of beads on a string, which will then influence the structure, which might then cause this protein to become less stable. And what can happen then is you can get what is known as an aggregate. So this is where the protein unfolds and forms these huge clumps that then inactivate the protein. Okay, so we can see a good example of this every day at breakfast, assuming we have an egg, of course, yes. So egg white, when you heat it, so heat is a very common stress, it goes and becomes white. The protein aggregates. Here's the egg white albumin, which forms the majority of that white. And you can see here that this is folded, it's interacting with water, you have a nice liquid, clear, smooth white, and then it becomes much harder and solid when you subject it to heat, because essentially you've unfolded that protein. So the question is, well, if we can cook an egg, can we uncook an egg? Okay. And the answer, according to some Australian and American scientists, is yes, we can. And so, I mean, that's a fantastic phenomenon thing, that you can actually uncook an egg. You can go from this yolk and you can take it back. Okay, now admittedly, you have to do some rather dramatic treatments. So you have to chop it up, you have to put it in a strong chemical environment, you have to treat it with enzymes, and then you have to put it in a machine and spin it round and round and round and round until finally it returns back to that form. Okay, so that's great. That's all great and well, and again, it's a phenomenal achievement. But what if that protein is sitting in your brain? Okay, I'm pretty sure it's going to be a hard sell to the ethics community if you're going to try and suggest that strategy that I proposed that they use to uncook this egg. So that's fine if you're looking at biotechnology processes with proteins that are in isolation. Probably, no matter how much you dislike somebody, you wouldn't subject them to that treatment. But it doesn't matter, because fortunately, the cell has its own system. It has a system that has been passed down through evolution. It's in all organisms, practically all organisms, and these are the molecular chaperones. Okay, they're a class of proteins, and essentially what they are little protein folding machines. Okay, so they will capture that new protein. Okay, they will help it to get to its mature form. All right, if that protein should encounter some stress, they will then help it to return back to that mature form. So essentially analogous to the concept of a social chaperone, just for proteins, to capture a newly formed individual, to monitor it closely ensure it does not participate in any unwanted side reactions, and allow it to arrive intact at maturity. And so hence the name really well describes what these proteins do. And so essentially what they allow in this scheme is for these inactive ones to be returned to support this process of achieving a correctly folded one, and really to then prevent this one. So they act as sort of stop-go signals at these particular points. And there's a whole bunch of them. There's lots of them. They form a massive network where they talk to each other and they interact. And their ability to support this protein folding is really critical, critically based on that network and interactions. And because they are so crucial for protein folding, and because proteins are so important for life, chaperones are often essential or vital for survival. And they really do cut across multiple spheres of biology. So we can see how chaperones function in non-communicable diseases. Okay, so diseases like cancer, diseases like dementia are intimately linked with chaperone function. In the case of cancer, cancers actually have chaperones have a cancer promoting activity, because mutations that drive cancer, which should be degraded by that dustbin in the cell, are actually stabilized by these chaperones. And so you have an inadvertent situation or an unintended situation where a system that is meant to help you survive transient stress becomes something that can sustain a cancer phenotype. And there you would look at inhibiting chaperones as a potential therapeutic target. Dementia is the other way around. Dementia is essentially associated with loss of chaperone function as we age. Okay. Unfortunately, we know that everything slows down as we get older, except perhaps time. And so it really is a case of chaperones become less efficient, and hence you get these massive misfolded aggregates within your brain, and that then leads to death of tissues, etc. But chaperones are also important in infectious diseases. Okay, so they're critical for parasites, bacteria, viruses to be able to survive in host cells. Many of them will have chaperones of their own, these pathogens. And if they don't, they will hijack the chaperone systems of the human host. But chaperones also have applications in the context of the environment and evolution. We were fortunate enough to participate in a collaborative study to look at chaperones in the coelacanth. And this is an ancient organism that hasn't really changed much. And it has a conserved set of chaperone systems that are very similar to other animals. But because these proteins are also linked to cell stress, they can be used as monitors for things like increasing heat and climate change, and for monitoring pollution. And we have a new project that's up and coming called the Hot Birds Project, where we're working with a collaborator at UP, where they found birds that have a substantially higher body temperature. And they think that this may be critical to them surviving in the current climate. And then, of course, there are many other biotechnological applications of chaperones, particularly in terms of making protein products, etc. So now I will move on to the last section of my talk, and I'll tell you a little bit about what we do and a little story of one chaperone that we've looked at in the context of cancer. So essentially, our work cuts across two aspects. So one, we look at fundamental science. So we want to ask the question, why do these proteins work in this way? And we want to add to the knowledge base of that. And through that, we use mostly genetic approaches, which I'll talk about now. And then we have a drug discovery angle where we then hopefully apply the information from the fundamentals into trying to find new therapies. And I point out now, again, we're using a genetic and a pharmacological approach, because if I show you this pipeline, which is a sort of representation of how you would filter down entities if you were doing drug discovery, you would start with your basic research, and then you would focus down into a single drug. And we always think of drugs as pills, but we really are in the era of genetic medicine and gene therapy. And so it's amazing to see how many of the tools that we use are now being adapted for actual therapeutic use. And I'll just show you two examples of, this is obviously the CAR T revolution for the latest sort of technology and gene therapy for cancers. But also this drug, they all have unpronounceable names, so I'll just hope you can all read. For muscular dystrophy is a gene therapy, and that costs $3.2 million. Luckily, you only need one treatment. Phew, yeah, panic over. So our work has traditionally worked, as I said, at a cell-based level. So we work in cell line models. And really, when we started, we were taking more of a phenotypic approach. So we would come at our drug discovery angles from screening, say, libraries of natural compounds, looking to find ones that had particular activities, and then trying to describe how they worked, and then hopefully we could understand their function and funnel them into drug discovery. And we worked with this, obviously, with Greg, but also we had an interesting collaboration with Denzel Bukes, and a lot of this was what formed Joe Delamere's PhD. We also got ourselves into an interesting situation. So we've had some in the later years, some better interactions with the media around our research, but this was our first one, where we arrived to work to find out that apparently we'd cured cancer. Now, I understand it's only the George Herald. No offense to those of you from George. I'm not sure how wide its readership is, but it was sufficiently widely read to cause much mirth and panic in us, and we had to have a strategy. And in the end, it turned out quite well, because we were able to get ENCA to come, and we could do some damage control and whatnot. But I'll never forget being in the department and Mike Davies Coleman coming in and saying to me, "What are you still doing here?" I said, "What do you mean?" He said, "Well, considering you've cured cancer, I thought you could at least take the afternoon off." And I've never forgotten that. So now we never say we're curing cancer. We never, ever say anything like that. And obviously we are very much at the forefront of, the beginning of that pipeline. So even the stuff that we're working on is really early days. And then sort of over the years, we've then sort of gone from this, using these chemical inhibitors, into applying genetic tools and really trying to establish in the lab cutting edge technologies, using technologies that allow us to genetically manipulate cells in order to ask questions about how those proteins function. And we've also moved from a primary focus on mammalian cells and cancer cells into looking at the systems in some viruses and bacteria. And I really, again, have to say that this type of technological revolution for our lab has really been driven, again, by the nature of the students that I've been fortunate enough to work with, because it's really taken a lot of development. And I think the fact that we do not like to let the techniques stand in the way of a good story. We like to be able to develop these new approaches and try them. And perhaps we are slightly foolhardy that we think, oh, it's easy, we'll just do it. So I really do appreciate their willingness to do that. Okay, so let me tell you a little story about one cancer angle before I end. So cancers are interesting, again, from a chaperone perspective. So I said that in this case, what you have is a system that's meant to help you, actually working against you. because cancers are driven by mutations. Okay, so they're driven by changes in sequences to proteins that then cause those proteins to have a different function. So here you would have a protein, it would perhaps have a mutation, and it would acquire a particular cancer-related function. Now, what should happen is in the cell is that should be less stable than this one, and so it should degrade. But along comes your chaperone, stabilizes that, and so you have a pro-cancer phenotype. And so if you can understand this and you can find inhibitors, then you have a strategy to block cancer. And the reason that this is attractive is because cancers are often not driven by only a single mutation. Okay, so if you look here, here's an example of, say, a cancer network. Here are all my mutations in red. If I'm going to want to target each of those mutations, I'm going to need 4 different drugs. Okay. But for these targets that are stabilized and engaged by chaperones, you can theoretically, through one chaperone inhibitor, simultaneously destabilize 4 cancer-promoting mutations. And so that really is the power in terms of that combined effect linked to the need to stabilize these proteins. So we've done a lot of work on a protein called the HSP70, HSP90 organizing protein, or HOP. Again, this is, I think, Greg's first love. And we were fortunate enough to work on it and to carry on doing some of that work. And so what I have here is just some data to show you that HOP is linked to cancer. So here are just some examples of images of tumor and normal cell samples or tissue samples. And the intensity of the brown color gives you an indication of the level of the HOP protein. So you can see across those vastly different cancer types, you have substantially more brown staining in the cancer tissue compared to the matched normal tissue. And so that tells you, for whatever reason, this protein's levels are being increased in the cancer scenario. If you also look at survival, okay, you can see that people who have high levels of this protein tend to survive less, or they have a shorter survival than if you have low levels of this protein. And then we can see across a wide range of different cancers, you don't have to worry about what they are, but that there are a series of alterations to these proteins at the genetic or genomic level. So this really convinced us that this protein was something worth looking at in terms of seeing whether we could correlate it with the cancer phenotype. Okay, and so here we had to develop these genetic tools. And so again, we used 2 tools that were, again, won the Nobel Prize in 2006 and then the Nobel Prize in 2020. So we used a CRISPR, which are like molecular scissors. You can go in and cut genome and get rid of a particular section. Or you can use RNA interference, where you can just reduce the levels and you do it in a temporary fashion. So you can let them come back. And then we also used this approach called overexpression, which, to the best of my knowledge, nobody's won a Nobel Prize for. And that's where you artificially elevate the levels of a protein. So you can have this yin and yang situation. You can ask a question, what happens if I increase the levels, or what happens if I totally reduce them or completely knock them out? And so we did this. We created a series of things for HOP. And then we did what we assessed a series of cancer hallmarks. So cancers have what are known as hallmarks. These are characteristics that define the cancer phenotype. And so you can assess whether the presence or absence of that protein is linked to supporting these phenotypes. So, for example, migration is one of the main ones, and you can actually study this in cell culture. You can watch cells migrating. And if we look here, if we look at an example of, here's the control, and you can see between these 2 black lines is a wound that we've made in this cell layer, and we allow that wound to then repair itself by migration over time. And when we do that by depleting the levels of HOP, we can see that we have, for starters, a much more erratically shaped wound. Okay, so we have an effect in terms of the direction that the cells are going. And we can see that that wound is much greater after the 24-hour period incubation than if we have HOP present. So this suggests that HOP is a driver of cancer cell migration and then somehow is supporting that phenotype. We also looked at proliferation, so the ability to grow. And here we can see, here, if we reduce the levels of HOP, we slightly reduce the ability of the cells to grow. And if we increase the levels of HOP, we increase the ability of the cells to grow. But really, it's not, we still see some growth. So the effect on cell proliferation is fairly minor. And then we look at it another way. So we look at the ability of these cells to survive long-term, a long-term survival assay when they're in a low number of cells. And we can see again, if we increase the levels of HOP, we increase the ability of these cells to survive long-term. So all of these three factors together suggest to us that HOP is driving a cancer phenotype and therefore is a potential candidate for inhibited development. Around about that same time, we came across a new and quite interesting cancer model. Okay, and this is a cancer model that's an intersection between infection and cancer, because it's caused by a virus called Kaposi sarcoma-associated herpes virus. And it causes a cancer described by this Austrian medic called Moritz Kaposi. It causes a cancer called Kaposi sarcoma. And this cancer is really a cancer of Africa. This is within the top 10 cancers within our country, and it's disproportionately prevalent in Africa. And that's because we have very high levels of the presence of this virus, just occurring naturally. They do very little harm to most people, but in certain conditions, in certain genetic backgrounds, and then in the context of immune suppression, you start to see the manifestations of the cancer associated with this. And so this really is an important disease in terms of our continent. And to the best of my knowledge, we're only the second group in the country to start working on this. So we really see that this is a really great opportunity to start playing a role for this important tumor. And it's a really interesting one because this virus lives inside B cells. So it infects your cells. B cells are one of the cells of your blood. And it lives in there. It goes in there and it attaches itself to your DNA. And then it just sits there quietly, biding its time, replicating as your cells replicate. But when you get stress, okay, it causes this virus to do what's known as reactivation. And then it starts to produce these viruses that then go on to infect endothelial cells, transform them, and give rise to the tumor. So really, we know what causes this cancer. And so if you know what causes the cancer, that's really great news because you can now intervene. And this is really the focus of this large grant that we've got from UKRI, and it's going to be a large part of the work that we're doing over the next couple of years. We've also been able to really revamp our tissue culture facilities and recruit some collaborators to work with us on this project. And very excitingly, so in terms of chaperones, viruses are, I mean, some of the most successful organisms, if they even are really organisms. They're, I mean, really a collection of genes, you know, and classified as dead, technically. But they are able, with such proficiency, to hijack cell systems. And that really is the way in which they're able to cause disease. And of course, they make proteins, and they have to fold their proteins. And so they use host chaperones. And so the aim now is to really find a drug that will leave the human proteins alone, but target the viral ones. And so we started with HOP, one of our favorite proteins, and we did, here's a little bit of staining. I hope you can see that. Hop is in pink here, and the virus, one of the virus proteins is in green, and you can see that they are situated in exactly the same place. So we believe that this hop is part of the process that this virus uses to replicate itself within the cell. And again, if we use our strategy, here we have the normal cells. You can see that's the amount of infectious virus you get. If you reduce the levels of hop, you reduce the amount of infectious virus. If you do the opposite, if you increase the levels of hop, you can increase the levels of infectious virus. And so we suspect that hop has an important role to play in this particular cancer. And obviously, we're really excited about taking a lot of the fundamental work that we've done in other cancers and normal cell types into this model as we go forward in the future. And then, of course, we're also linking that to trying to find some hop-based inhibitors. And we have one candidate here that is potentially interesting, and it seems to reduce the levels of virus as well. So that's really promising, and we're hoping that in the next couple of years, we'll be able to progress this and perhaps come up with a new strategy to treat this disease. So I'm at the end. Thank you. We've made it, only just. And in closing, I would like to say that, you know, I really feel very privileged to have been able to have been an academic. I mean, I'm talking now like I'm not going to be one tomorrow. Of course, I will. I'm coming to work tomorrow. Don't worry. So, but I think as a career, it's extremely rewarding. I know there's a lot of negativity about academia, but I've had the opportunity to meet amazing people, to travel to amazing places. I could do without the flying, but I really enjoy the new cultures and being able to meet interesting people. And what that also does is again, reinforces to us that things aren't always better on the other side, no matter what they might look like. And we have a lot to be grateful for. And we have a lot to, we don't realize how lucky we are until we go somewhere else and we see what people are doing under their conditions. And so I'm really, really keen on making sure that we're grateful and that we continue to work hard and carry on the great tradition that the group set up until now. So I will say thank you very much. And my last thank you is obviously to my family. Just to say thank you so much for all the support. My parents arrived by surprise at 4 o'clock this afternoon. So that was a wonderful, wonderful surprise. I wonder if somebody told them I had an important talk that I was preparing for, but anyway, let's not worry about that. And then obviously to my favorite output, Renee. It's not easy being the partner or to Gary as well, but it's not easy being the partner or the child of an academic. Sometimes there are many things that impede on family life. And so I really, really thank them for their patience and their support over all these years. And I'm delighted to say that Renee is probably the only person who likes experiments more than me. Thank you so much. Thank you.
It is on. Good. Welcome, everybody, and thank you for coming. I'm just scanning the audience. I see friends, I see colleagues, some of which I've told they should be wearing red and sitting in front. And there's, and your parents. Thank you. An inaugural lecture is a very difficult space. It's a difficult lecture. You have to sound intelligent, but at the same time, you have to be humble. It's also going to be the first time that you give a lecture to your parents. Adrienne, you've done a marvelous job. Thank you. It was sincere. We got to know you, who you are. I can remember you just after you're about to go on maternity leave saying, I'll only be on maternity leave for 2 weeks. Don't worry, I'll be back in the lab. But I said, please don't. So, yeah, you've dumbed down what you've done. You've made it extremely accessible to us. It's an abject lack of jargon. Thank you. I was disappointed with the benzene ring at the end. But you did a marvelous job. And yeah, we just want to congratulate you. You've now in the academy, you are a professor. And we look forward to seeing how HOP and chaperones are going to potentially provide us a cure for what looks like Africa's new waging war. Yeah. So here's your goodie. I've got no idea what it is, but you deserve it. Congratulations. And it matches. Lovely. Ladies and gentlemen, in the concourse, we have some refreshments and some nibbles. Please join us and we can congratulate Adrienne for her achievements.